happy that you're here with us this morning, this afternoon, and this evening, wherever you are zooming in from in the world. Today we have Dr. Ryan Blaustein, who has joined us to give our first talk of this semester. Dr. Blaustein is an assistant professor in the Department of Nutrition and Food Science here at the University of Maryland. He is a UMD alum, and he also received his PhD from the University of Florida and has extensive research experience in soil, environmental, and food safety spheres. His current research interests include microbial ecology, microbial genomics, food safety, and public health. And his talk will explore how shaping microbial landscapes within emerging food systems could impact the control and spread of antimicrobial resistance. So welcome, Ryan. We're so happy that you're here, and let me stop sharing so you can go ahead and share.
Thank you, Dr. Subkota, for the introduction. Thank you, Ash and Ope, for the invitation to speak with the Global Futures program today. I'm excited to talk a little bit about the work that my lab is doing over in nutrition and food science on the interdisciplinary topic of antimicrobial resistance in the food system. And I'll share with you some stories of a few of the projects that we're working on in this area. So first, before getting into it, just to give an introduction to my lab and our vision. So I'm a microbial ecologist. I study interactions between microbes, microbes in their broader environment, their response to environment, try to use ecological theory, genomics applications with applications to promote food safety and help to further food security. So ideally looking at our connection with the environment, with agricultural systems, with the built environment, with the goal of kind of driving and understanding microbial interactions to come up with strategies that will help us lessen chemical inputs and lead to a more sustainable future. Well, why is this important? From a practical standpoint, we can start with food safety. Everyone's impacted by it. In the United States, 1 in 6 people experience foodborne illness each year. There's FDA and many researchers have estimated billions of dollars in economic damages. And a lot of the root of this comes down to microbial diversity. For example, we want to know what's in our food, uh, um, uh, what can it functionally do, and then how is it changing? I think how is it changing in a way leads to why we see trends like this for increases in multi-state outbreaks, which is this data is a little bit old, but it does show that over a decade and a half, we actually saw increasing outbreaks in food safety, and makes us wonder if we've put a lot of funding into, or US government and other agencies have put a lot of funding into trying to combat this massive issue on our national and even global scale. Why do we continue to see this problem? And I think part of it is traditionally in medical industries and agricultural industries, a lot of us understand now that the anthropogenic influence on the environment has actually, yes, we've helped to try to improve food production, but we've done so in a way that might actually have a catch-22, in that we've changed the landscape to some extent where microbes are able to actually adapt to the environment, to be able to persist for longer times. Mobile genetic elements that mediate transmission of stress response capabilities, for example. And it goes beyond just our overuse of antibiotics in medical industries, in agricultural industries to improve meat yields, but also this is, well, in a way, in some ways impacts, but also goes concurrently with other global problems as well, like rising climate temperature, rising temperatures, more erratic rainfall events as well. So my lab, we have several projects that we'll talk about today that both follow the same general hypothesis, although they focus on different themes in this area. And that working hypothesis is that understanding the response to environment of particular foodborne pathogens that are involved in this broader food safety issues has potential to improve the system. Really understanding those interactions, understanding genetic signatures that may underlie this particular response, understanding novel function can help us to improve genotype-based risk assessment, expand abilities for molecular surveillance, like such as with shotgun metagenomics, and then maybe come up with new strategies for control as well. If we know how we're shaping the environment and how microbes are responding to it, then maybe we can come up with more sustainable ways to do that going forward. And over in nutrition and food science, I will want to highlight that this type of work and understanding interactions within it really is broader than just the food safety system. But when we think about food science, microbes and microbial communities, and even antimicrobial resistance plays a really important role, not just for consumer health, but also for crop production. We think a lot about human pathogens, but actually plant pathogens may cause more of a problem when we think of food security issues. 20% of crops grown globally are lost to plant disease, for example. And then, of course, microbes play foundational roles in food spoilage as well. So my lab does, today we'll talk mainly about food safety, but then also I'll end with some of the themes of new directions that we're exploring related to crop production and quality. And I think that this work fits certainly within the themes of your program, the food at the nexus of food, energy, and water, through the One Health scope. And antimicrobial resistance is an important attribute of microorganisms as they are adapting to the environment to be able to overcome stressors, persist for longer periods of time. If those types of functions and features can get to things that make us sick or things that can make plants sick, then we actually have a problem. So the work that we'll talk about today, I'll tell a story about a project that we recently completed and have ongoing future directions related to urban farming and how growing organically can actually promote healthy microbial diversity within the soil, within the food system. Then some of the bioinformatics work that we've been doing related to understanding how foodborne pathogens are adapting along the agricultural continuum to consumers and how we might be able to use that information to help improve molecular surveillance. And then I'll conclude with an introduction to some of the work that we're starting on crop microbiome and its role in food quality and potentially biocontrol of plant pathogens as well. All right, so for that first project, uh, I think everyone here is pretty well aware of the benefits of urban agriculture and growing organically and local food production. Of course, urban ag has numerous benefits from helping support food and nutritional, combat food and nutritional insecurity, engage communities, promote environmental sustainability, so local production, less distance traveled from farm to fork, less fuel, less emissions, um, uh, as well. So urban ag is very important for many reasons, but it's actually a bit challenging to set up a farm. And grow produce that is of high quality because the soils that we're growing on are often depleted in organic material or maybe contaminated with pollutants that come from emissions, heavy foot traffic, creating soil compaction, loss of organic material. So when we think of urban agriculture, it's important to remember that we need to really change that microbial landscape, change the environmental chemistry of the foundation of the system to be able to grow produce effectively and of high quality. So my lab wanted to ask, or well, we asked this question and we conducted a project back in 2023 to understand microbial diversity at the foundation of these systems here in the Washington, D.C. area. So we went to different urban farms and community gardens here in greater D.C., from D.C. to PG County and Montgomery County. And we wanted to understand how farm management, how land history are impacting local environmental conditions, and then how that correlates with microbial diversity that is at the foundation of this system. And we did this with two different scopes. One, we wanted to do culture-dependent assays where we would actually quantify what are the total bacterial load, what's the phenotypic diversity of antimicrobial resistance within this system. And then we also did we used molecular technology to look at the microbiome as well and whole genome sequencing of isolates to understand what is the genotype and the genotypic diversity therein. And really, the idea is we want to know how does this microbial diversity reflect farm operations, different environmental conditions, and then potential contaminants within the system as well to inform strategies to promote healthier soils. And this expands from just soil health to food safety as well. When we think of uh, 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 pollutants in the environment, we could also be selecting for undesirable traits within the microbiota that we're consuming with our produce. So we went to 7 different farms in the DC area. We returned to one of the farms twice. This project was spearheaded by PhD student Qingyue Zheng in the top left of the pictures here. And these are and some other undergraduate students that played a really important role in helping take our samples. So we collected soil at each of the different sites. We collected soil in the root zone where the crops are actually growing, so where the amendments have been used. And then we collected produce themselves, so different leafy vegetables, kale, lettuce, chard, and cabbage. We brought these samples back to the lab, processed them for microbiological analysis as well as molecular analysis to study the microbial diversity and antimicrobial resistance within the system. Again, as a factor of amendments used, local conditions, land history, which we expected would be largely heterogeneous as urban farms and urban agriculture can vary widely from site to site. So what did we find? I think one of the key findings here, and the first finding is actually quite expected. When we're amending the soils, we see an increase in total bacteria within where we're actually growing the crops. So what these box plots are bacteria plate counts on the top row and then percent tetracycline resistant on the bottom from each of the different farms, community gardens that were surveyed throughout summer 2023. The purple boxes are bulk soil at each site. So these were like a site control. And then the green and yellow colored boxes are different plots. So again, chard, kale, lettuce, and the different cultivars that were grown. You can see pretty clearly on that top row that relative to the purple boxes, there's an increase at the farm level across all plots. And some plots specifically more so than others in total bacterial load. So about one order of magnitude, we put soil amendments into the ground. We're already having a much larger bacterial population density. On the bottom panel, and this was not necessarily expected, but a very interesting result, was that the proportion of organisms within that within the total bacteria population, the proportion that were resistant to tetracycline actually decreased at most of the farm sites. And you can see the opposite where the purple boxes are significantly higher than some of the other green boxes in that bottom row. So what this tells us is that amending the soil actually, as we understand, promotes bacterial activity in some way. Now, it could be we're putting new bacteria into the soil, we're stimulating native bacteria that are already in the soil, or even we're diluting out the maybe harmful bacteria that might already be in some of these soils that could have been responding to contaminants in the soil. But what we see is by promoting soil bacteria activity, we may also be reducing burdens for antimicrobial resistance. We also did screen for food safety indicators like E. coli and coliform bacteria. While we got there were similar trends like you see for the TET resistant for coliforms, though E. coli were often below detection range. So food safety was more of a low risk at all of these sites to begin with. But what we do see is that antimicrobial resistance is also reduced in a way. So setting up urban farms can actually help with the spread of this problem into the local food system or into the food system in general. And this was important not just for resistance to tetracycline, but the fact that a lot of the organisms that were resistant to tetracycline we saw were resistant to a whole panel of clinically relevant antibiotics as well, azithromycin, ciprofloxacin, and so on. In fact, about 70% of the isolates with unique morphology from the different sites that we recovered that were resistant to tetracycline were multi-drug resistant or resistant to 3 or more antibiotics. So trying to reduce that burden we can confirm is a pretty important scope. So how do we reduce that burden besides just setting up more urban farms? Well, we can think about the environmental conditions as well. Here's at the farm level across the different sites that we went to for our survey, an interesting correlation between soil pH, which is in a tight window between 6.5 and upper 7. So not too broad of a range, but there's an important correlation between drug-resistant bacteria within the cropping soils and then with soil pH. So potentially controlling or targeting a zone that might be mildly below neutral, which we know is actually important for nutrient bioavailability for the crops themselves as well, could also have added value in supporting an ecology that may favor less antimicrobial resistance. Though I will say this is correlation, this is not causation. So going further with this story, so we talked about so I guess all this data to this point is what were we able to culture? What was the phenotypic drug resistance, um, or what were the drug resistant phenotypes? Now we're stepping into the using molecular technology to try to understand this a little bit further as well. Um, from site to site, there were differences in the microbiota present within the soil and then also within the produce that we had surveyed. We used 16S sequencing to characterize bacterial communities, ITS sequencing to characterize the fungal communities. The two left panels focusing in the soil, the open circles represent bulk soil, the closed circles are soil in the root zone. So where amendments have been used, where we're actually growing produce. So there could, again, there's a new biology, new chemistry within those soils because it's a mix of organic material with the native soil. And then there could also be a rhizosphere effect as well. So I will point that out. But you see a pretty distinct microbial community when you look from what's actually there and how we're actually managing the soil. And then there are key differences from site to site as well that do reflect some of the properties or the variables that were targeted in the survey. So, and I'll get to that in just a moment. But I do want to just pause to appreciate that each site there's a different clustering within the respective microbial communities. And this extends to the produce. So on the far right, the plant bacteria, things that were actually on the leafy greens, do cluster by site as well. So there's actually microbial signatures. If we think of we're eating a salad from a farmer's market, there's microbial signatures from the farm at which that salad or that lettuce or kale had been harvested from that could be important for us as consumers and maybe even plays a role in food quality. So some of the factors that we were able to, with this survey, distinguish as linked to the produce microbiome and the soil microbiome where we're growing produce in urban agriculture systems, of course, is very site dependent. And site is confounded directly with the specific soil amendments that were used at each site. We do have within site variation, which not shown here, but as well, some sites use different amendments, and we were able to capture differences in their microbial diversity. But from the different sites, we were able to also evaluate the effects of different irrigation sources, which are shown. An example of that is shown here. Region, as in like DC versus Montgomery County, PG County. How old is the farm? Because a lot of these farms are pretty young in operation. But all these factors do play a role, and they are confounded in some way. So in the future, like if we want to optimize the microbiome, we would need to try to better understand those connections. I'll focus on irrigation source for just a moment to emphasize that we had surveyed 7 farms, 4 of which had used city water and harvested rainwater, 3 of which had used either pond water or well water. So we have natural water compared to treated water. And within the soil, within the produce, and well, within the soil, we see important impacts of irrigation source on not just bacteria, but also fungi. And then on the produce, we see that as well. The PCOA here is showing differences in the microbial profiles, the bacterial communities in the amended soils. And there's a pretty sharp distinction between sites that had used one or the other. Again, this is 4 farms compared to 3 farms. So it's relatively small sample size, but this is kind of interesting to see that right here in DC, what we're putting into the system is actually shaping the microbial ecology as well, and beyond just the soil amendments. So on the topic of water, and not to go too far on a tangent, I do want to mention that there is important within site variation, not just based on the soil amendments, but on the water, where we're actually, and if we're trying to do a survey to sample and understand why that's important, we have to consider importance of sampling strategy as well. So this was another study with some colleagues out at USDA. This is not an urban farm. This is actually at the Y Research Education Center affiliated with UMD. We looked at the agriculture irrigation water that was sampled at different depths, at different times of day, and we saw that both depth and time of day had an important impact, or well, on the microbial diversity that we would capture when we would employ those sampling events. For example, if we sample in the morning, we have a lower microbial diversity than if we sample in the afternoon. And of course, that could be because things are growing. There could be inactive cells maybe a little bit later in the day. There could be some kind of a bloom as things warm up. But understanding the ecological dynamics, even within the system at the fine scale, can be important for understanding risk related to food safety. For example, at the 12 p.m. slot here in this survey, we can see a correlation between microbial communities and E. coli that was actually cultured within this system as well. So the microbiome could have understanding the microbiome and microbial interactions could have implications for being able to predict food safety risk within sites as well as across sites, from the water to the soil. So for this first part of this presentation, to conclude, within urban farming, urban agriculture systems, and thinking about the spread of antimicrobial resistance, of course, microbiology is very important for growing fresh produce, changing the landscape, changing the soil properties, adding organic material. We've known for forever that's actually that we need to do that to grow produce, and now we have a better idea of what are the microbes that are involved. And we'll be following up with another paper that will expand on those themes. Um, Soil amendments and really promoting urban agriculture can also have value added in controlling potential risks related to public health when we think of antimicrobial resistance. If soils are often contaminated with heavy metals or even antibiotic residues or other chemical residues, and the resident soil might have higher levels of drug-resistant organisms, adding organic material, creating a healthier environment certainly could have applications for controlling the broader spread of antimicrobial resistance as well. And this is important to consider that in that more than 50% of the world population, billions and billions of people, live in or around cities. So setting these systems up is going to go a long way to have a large, far-reaching effect. So in the future, my lab's going to continue along these themes, try to better understand applications to mitigate antimicrobial resistance, such as that association with soil pH, maybe other soil conditions as well. And not just to promote food safety, but also thinking about food quality, growing organically for value-added products. So the second project that I do want to talk about is going a little bit outside of the Maryland area. So we focus on urban farming in and around Washington, D.C. Now, this project is sourcing publicly available genomic data from all over the world, from North America, from Asia, from Europe, and other locations as well, to understand genomic signatures of foodborne pathogens as they vary across different sources along the agriculture continuum to consumer, and then across scale. Again, that working hypothesis, how organisms are responding to their environment, local environment conditions, can have applications to promote food safety, such as through improving or understanding adaptation, understanding ways that might be more effective for control, and potentially identifying markers for genotype-based risk assessment. So in this project, which is supported by USDA NIFA, we're focusing on foodborne pathogens that are relatively understudied, but disproportionately impactful to unique products, such as Chronobacter sakazaki, which has been responsible for recalls to powdered infant formula, Campylobacter jejuni, uniquely associated with poultry, and then Lacera monocytogenes, which has a broader niche range from dairy to fresh produce, but its unique ability is to withstand lower storage temperatures. And this work, so we use computational tools to try to navigate genomic diversity across scale, across sources. And this project is being led by a wonderful postdoc in my lab, Dr. Marie Gao, who has been working on a workflow to navigate large-scale, publicly accessible data, starting with Chronobacter. We'll talk about the Chronobacter example today, but then we'll eventually get into Campylobacter and Listeria as well. The general workflow is we go into larger database archives of genomic data. We pulled those genome sequences as well as the metadata associated. One of the challenges with doing this, of course, is that the metadata, as these data are collected from all over the world, from different research groups, the metadata are not entirely organized or reported the same way in a consistent basis. They're very powerful, but it needs some fine tuning. So Myri developed a method for QC of not just the genomic sequences, but QC of the metadata as well. Actually using ChatGPT, well, not ChatGPT, GPT Playground to model and parse the metadata into different source groups, be it food, environment, clinical, and then subgroups respectively. So for Chronobacter, for example, powdered food, powdered infant formula, flour, these are important food products that this has caused major problems for, but it's been isolated from other foods as well, like spices or nuts or other dried foods or dried fruit, and then other foods as well. So we take the metadata, the genomic data, build up what we call a pangenome. So annotate all of the genomes, understand what are the different functional gene clusters within the whole system, even further characterize specific features such as antimicrobial resistance or virulence or even metabolic pathways, and then use machine learning to try to understand the associations between these features and then back to that metadata. So then we could say, oh yeah, Chronobacter from powdered infant formula is more prone to carrying gene X than Chronobacter from isolated from soil in some system. And this type of workflow can lead to, you can ask many questions with the data, but the idea is what is a standardized, we don't really have a standardized way to do this yet, so we've been working on that. So for Chronobacter, the metadata model that Myri worked on had a very high accuracy in the reassignment of source type, so environment, food, clinical, had a strong accuracy and precision to a lesser extent in those subcategories as well. So it's important to take that into mind when we think of how do our genomic data actually correlate back with these metadata. So the pangenome for this group, it really wasn't too big of a data set. It was over 2,000 isolate genomes that we had looked at. But the idea in starting with Chronobacter is we can then upscale to something like Listeria, which has 60,000 different isolates. So how can we automate the system here at the small scale and then expand to broader foodborne pathogens as well? So she identified the gene clusters associated with this pangenome, and within those gene clusters, which genes had homology, sequence homology to virulence factors as well as antimicrobial resistance. Because again, we're going back to the question of what makes particular strains unique under different circumstance, be it from different source type or from different geographic region. Focusing on source type, I'll point out that, so Chronobacter, for example, this is all the same species, Chronobacter sakazaki. So it's expected that the total gene content per genome is going to be pretty universal. Though we do see at the broad scale, compared to isolates from the food facility versus in food themselves, there's actually significantly higher genes per genome. So then we start to wonder if there's 4,100 genes in these genomes on average, and then 4,150 genes in these genomes, and this is significant. What is the difference? Are these plasmids? Are these acquired functional features that could be undesirable? And then when we look further into, if we look more specifically at, again, the data set's very large, so we can ask different questions at the genes per genome of different food types, we see that powdered foods, of course, carry higher number of genes per genome, slightly but significantly than other dried foods and other foods where this organism has been isolated from. So again, we know that this organism has caused problems to the powdered infant formula industry, flour industry, but not really other foods per se. And we know that it responds very well to low water activity. But what are the other features? What are the potential risk factors that could be associated relative to, because it's an environmental organism, it could be present elsewhere as well, too. So this is where it gets a little bit fuzzy. The genes, so I'll try to explain this phenomenon. So the two Chronobacter that are the same species have the same core content, but they have different accessory content. And those accessory features are functional features that we think might reflect, well, we know they reflect the unique history of that organism in space and time, but they may associate with particular conditions of that organism as well. When we compare isolates that came from clinical specimens, different food facilities, so environment, versus actual food, powdered food, it's hard to distinguish necessarily what are the traits unless we use a more complex model. So using on the PCOA on the left, you can see there's a low effect size, but there's significant clustering. But then in the middle, you can see that when we try to use a random forest model, we can pick out the jigsaw pieces that comprise a particular genome that is coming from a respective environment. And there's very high accuracy and precision in doing that. So what are some of the genes that are involved in distinguishing isolates that are coming from different sources? Well, a lot of them are actually hypothetical proteins, so genes that we don't really know the functional characterization all too well. Again, it's a non-model organism, but there are some genes that have been annotated more broadly. The heat map here is showing the percentage of isolates reflective of the unique source clinical environment food. So blue is, if it's blue, it isn't found in any of that. If it's red, it's found in, or 100% of that. You can see some different patterns here on the heat map. The bars here are showing um, the importance of different gene clusters in making these key distinctions. And then they're colored based on COG category or metabolic feature. There's a lot of gray, a lot of function unknown, but I have highlighted also which are the genes that have homology to virulence factors, and actually quite a bit of them that seem to be important too. And a lot of them are actually hypothetical protein. So using this method, we can start to dissect what are some key components of the genome that might be able to distinguish Coronabacter as it is derived from different sites or different sources, and understand that these could actually be virulence features that we don't really know that much about yet. This example is a little bit more clear when we try to compare powdered food versus other types of food. There's also distinctions within the genomic profile. You can see, again, the effect size or R squared is relatively low, but it's significant. There's a little bit more clustering in the PCOA. The random forest model is very powerful in being able to distinguish, did this isolate come from a powdered food sample compared to other types of food samples? And here we have a little bit more clear and annotation of some of the features that might be distinct when we try to look across different food sites. So again, the white is like about 50%, the red is about 100%, and blue is about 0. So you can see down the heat map that powdered food compared to other types of food or dry foods, there are some distinctions in the functional pathways that are encoded in the accessory genome. And I've highlighted again virulence factors. And then if there's an A, it also has homology to an antimicrobial resistance gene. So I'll focus on anything here that says COP something or CUS something that has to do with copper tolerance. So uh, going back to the story of how is antimicrobial resistance involved, being able to efflux other chemical stressors, maybe metals that are actually used in food processing, or Coronabacter appears to carry at a higher rate these types of genes, and they could be plasmid associated. We need to do a little bit further analysis as associated with powdered food versus other foods. So it could represent how this organism might be adapting to the food system. Now, some of these genes might actually play a role in if we were to improve our risk assessment frameworks. If we wanted to focus on, well, is the pathogen there? Does it actually carry this gene? What's the risk that it's going to persist? We could try to use this information to further those methods as well. So from the second project, I'd like to highlight that we've been uncovering genomic signatures of Coronabacter as associated with source and geographic location. The location data wasn't shared, but I will mention that actually when we look at the continental scale, the effect size is actually more powerful when we try to compare that than it even is source. So it is very important to understand the context from which things have been isolated, especially when we consider the global food supply or globalization of the food supply. This bioinformatics framework is our goal is to try to automate this and upscale this so that we can try to understand key features of other foodborne pathogens as they occur with their own unique lifestyles. For example, Listeria and Campylobacter, there's tens of thousands of different genomes. There's a few thousand different sources per se based on those metadata. So we would need to use AI, we need to use a language learning model to really parse these data. It's too complex to actually go through an Excel sheet and try to do that. But we can build off of what we've done with Chronobacter to try to expand it in this area. And then here on the bottom, this is that genes per genome phenomenon. This is Listeria, as it's been isolated from soil, from uh, um, milk-based products, and then also clinical specimens. You can see that there, although again, we're talking like 50 genes, 100 genes on average difference, but that can actually be quite significant when we think about the implications from a functional perspective. So this study, of course, our findings will have applications to improve methods for source attribution, and this couples or pairs pretty well with what's already done from a phylogenetic perspective in common practice that FDA uses. It could have impacts on a genotype-based risk assessment, and then also new targets for molecular surveillance. I'll mention we're also trying to benchmark metagenomic tools for detection of diverse foodborne pathogens, because one of the limitations when we use metagenomics is that a lot of the sequences are actually not characterized. And part of that is, well, we don't necessarily know what they belong to, what they do. But through this type of framework, we can say, oh yeah, but these hypothetical proteins are actually really important. They should probably go into that type of database if we're trying to detect Chronobacter, for example. And then lastly, without going into the data, I did want to introduce another project. So we've talked a bit about food safety and antimicrobial resistance, soil health and antimicrobial resistance. And now I want to talk a little bit about um, uh, sustainability from the perspective of food quality, and actually not resistance to human pathogens per se, but resistance of plant pathogens. So in a AGNR Fearlessly Forward project that I'm leading with collaborators from plant science and extension, and then others in my department of nutrition and food science, we're working on viticulture in Maryland. So fruit production, small fruit production, grapes in particular, going from farm to glass, not farm to fork, when we think of grape growing to winemaking. This project has two objectives. The first, we're trying to model the climatic conditions across the state as driving different yields and quality of the fruit themselves. So how is the vine maturing as it goes from budding to flowering to setting, and then actually the grapes enlarging, ripening, what is the sugar content, and so on. How is that reflecting changes in temperature and soil moisture and leaf wetness? And Dr. Diana Cochran in Plant Sciences is leading that piece. And then in collaboration with some other faculty, we're doing a microbiome piece on this as well, where we're trying to study how is the microbiome changing within the entire growing environment. So from the soil to the leaves and then the fruits themselves, what are the core microbiota that associate with grapevine? Could understanding who's important have applications for biocontrol? Can we try to promote that microbial diversity rather than intensive chemical sprays? So for the grape industry, this is among probably top two in terms of agriculture products that actually receive most pesticide treatment. So there's a very intensive spray schedule for fungicides and to control disease. And some of the sites that we've worked with will have important problems despite the use of seemingly effective spray strategies. So anything that we can do to try to be more effective and reduce, again, chemical inputs to improve the product will be important. So we're trying to look at the microbiome for some ideas to be able to do that. And I think that this work extends actually to food processing and quality as well when we think of actual winemaking, because potentially some of the yeast that could be used in biocontrol could also have application in fermentation. Maybe there's certain flavor attributes that that could be controlled in the nutritive value of the food product. So what we've done so far, we've gone to 5 different research sites in the state representing Western Maryland and Southern Maryland and the Eastern Shore and Central Maryland. And we've set up weather stations to be able to collect that climate data, and we can model that. And I know the industry is really excited about these partnerships. And then we've also gone out and really just sampled the environment, what's going on in the leaves, in the roots, and on the berries themselves, the berries that are used to make wine. These are some pictures from the recent growing season and students in the lab this summer that have been very important to supporting this project. So we're excited about what's going to happen next. And then in the lab, we're isolating um, uh, yeasts from the grapes themselves, maybe trying to use like a native yeast or a native fermentation to Maryland, compare that to what could be done from an industry perspective or commercially, where Saccharomyces is very well developed for this industry, but it would be kind of interesting to see how we can alter flavor profiles and food quality when we think about other microbes that could be involved. So we're doing this, and we're doing this is like small scale fermentation. We can try to do this high throughput, and then eventually we'll get into maybe larger production. But more details to come in the future. I just wanted to introduce this project because I think it's really fun and again, relevant to the theme of microbial diversity importance at the foundation of the food system. And as far as antimicrobial resistance goes, because we're spraying so many chemicals to grow grapes, trying to mitigate risks associated, could have far-reaching outcomes. So I'll conclude with some of the same themes that we talked about in the talk. Organic production, particularly urban farming, can help promote a healthy microbial diversity, not just in the soils, but maybe on the produce themselves. And that's important to consider when we think of us as consumers. Are we eating salad that actually has less risk associated? And risk traditionally is, linked to like food safety indicators. But when we start to think outside the box or think from a One Health perspective, we can also think of antimicrobial resistance. We have computational research that helps navigate how foodborne pathogens might be responding to the environment from farm to fork all over the world. And then lastly, we have some new directions where we're trying to understand the role of microbial interactions in the food system in quality of an important beverage product made here in Maryland. So with that, thank you everyone for your attention. Thanks again to the FEWtures program for the invitation to speak. I'd like to acknowledge the incredible members of the lab, postdoc, graduate students, undergraduate students that played a really important role in this project, and then some key collaborators that were involved with the different projects that we talked about today. So with that, happy to open the floor for questions. And again, thank you very much.